Enzyme reactions can be carried out in two ways, either as immobilized enzyme or free enzyme. So far in biology, we have done enzyme reactions with free enzyme, meaning that we would create a solution that includes the enzyme. Now we can either add the substrate to the enzyme solution, or we can take the enzyme solution and add the substrate. So in both these instances, both solutions are mixed and the enzyme is free floating inside of the solution and is able to bump into the substrate and catalyze a reaction. Now we can also do enzyme reactions using immobilized enzyme. So immobilized enzyme is where the enzyme is bound to an inert, stationary, and insoluble medium. Now inert means that the medium will not be able to react easily. Stationary means that it's not liquid or aqueous, and insoluble means that it's not soluble in water, meaning that it won't dissolve. So when we do this, we will pour the substrate over these stationary, inert, insoluble beads and then collect the product at the bottom. This is a very efficient way of catalyzing an enzyme reaction because no enzyme will be present in the product that we get from the solution at the end of it. We can also reuse the enzyme several times, and the enzyme will also have a greater tolerance of pH and temperature changes. So how do we produce an immobilized enzyme? So the first thing that we're going to do is we're going to add an enzyme to sodium alginate. Now, sodium alginate is a polymer that is often extracted from brown seaweed, and it has many uses. We can use it as a drug delivery system, in biotechnology, and in the food system. We're going to add our solution to a syringe, and in a separate container, we're going to add calcium chloride. We're then going to drop our sodium alginate and enzyme solution into the calcium chloride. This means that the calcium ions in the calcium chloride will replace the sodium ions in the alginate polymer. This will create cross-links between the polymer chain and form a gel shell around the enzyme bead. Afterwards, we're going to wash the bead and remove excess calcium ions. This is going to give us, again, an inert, insoluble, stationary bead that will allow our substrate to diffuse into the bead, react with our enzyme, and produce a product. So to use our immobilized enzyme, we can use a funnel or any piece of equipment that we can add the beads into. So we'll add our beads into the funnel and we'll then get our substrate ready. We can pour our substrate solution over the immobilized enzyme beads, and at the bottom we can collect our product. You can see here that the enzyme doesn't mix with the substrate at all, meaning that our product will be free of enzyme and no purification will be needed afterwards. The product can be used as is. Now, sometimes it's very important in enzyme catalyzed reactions to measure the rate at which the enzyme catalyzes the reaction. Now, we can do this in two different ways. The first way is measuring the amount of product that is formed and how fast this product is being formed. This can be demonstrated through the use of catalase. Now, catalase is an enzyme that breaks down hydrogen peroxide into water and oxygen. Now, oxygen is a gas and it can be collected using either an inverted measuring cylinder or a gas syringe. Now, in this experiment, what I did is I took some potato, and potato has some catalase in it. So I then put the potato inside a container that could seal, connected to a tube with an inverted measuring cylinder. I then have a solution of hydrogen peroxide. Now, it's very important in this reaction to keep the pH very constant. Catalase likes working at a pH of 7, so I used a buffer solution to keep the enzyme reaction at its maximum. I then removed all of the air from my inverted measuring cylinder to be able to capture the oxygen that would be produced. I then added the hydrogen peroxide to my potatoes and sealed the container really quickly so that no air escapes. So we can now look at our measuring cylinder and look at the gas being produced. We can see the liquid or the water level lowering in our inverted measuring cylinder. So if we wanted to calculate the rate at which oxygen is being produced here, we would set up time intervals and measure the amount of gas produced at each timed interval. We could then graph the results, and then if we work out the gradient of the graph, we would find the rate of the reaction. The rate of reaction in this example will be measured as decimeter cubed per minute or per second. The second way that we can measure the rate of reaction of an enzyme is to measure the disappearance of the substrate, or how quickly a substrate is being used up. So we can actually exhibit this through the use of amylase. Now, amylase is an enzyme that breaks down starch into maltose and glucose. Now, we know that we can test for starch through the use of an iodine solution. If starch is present, the iodine will turn from a yellow-brown color to a blue-black color. So in this experiment, I have my starch solution. I will then add my amylase enzyme to my starch solution. At this point, I would already start my timer to time how long my starch solution and enzyme has been in contact with each other. So here I have set up test tubes with different time intervals. The first time interval is 5 minutes, then 10, then 15, then 20, and then 25, and then I have 30 as well. That's a bit out of the camera view at the moment. So in each test tube, I add the same amount of iodine and water to create an iodine solution. So when setting up this type of experiment, I need to remember to try and keep all other variables that I'm not testing for constant. That is why I used the same temperature for the solution, the same enzyme and concentration of enzyme for the solution. My iodine solution is the same volume and the same concentration for all my test tubes. Now after 5 minutes has passed, I will add my starch solution to my test tube and then look at the results. So here I can see that my starch solution at 5 minutes still turns my iodine blue-black, meaning that there's still a lot of starch present in my solution. I can then add it to 10 minutes after 10 minutes has passed, and here I can see that the color change is slightly less, but it's still a dark color, meaning that there is still some starch present. After 15 minutes, I can add my starch solution again, and here I can see that the color change is very slight, meaning that the amount of starch present is at minimal. Then, of course, after 20 minutes, I can add my starch solution to the 20 test tube, and here I can see that not much of the color has changed at all. Then after 25 minutes, I can add it to my 25 test tube, and here I can see that literally no color change, and the same for 30 minutes, no color change. So I can see that there's a decrease in concentration from the 5 minutes to the 20 minutes, but between 25 and 30 minutes there was no starch present in my solution, and all the starch has been hydrolyzed into maltose and glucose. The problem with using color to determine the rate of an enzyme reaction is the fact that it is more quantitative than qualitative. So it's also very subjective, meaning that what I would see with my eyes, which is that the reaction was completed between 20 and 25 minutes, might change from person to person. So another person might say that the reaction stopped just before 20 minutes. and someone might say, no, it's between 25 and 30 minutes. But there is a method that we can use to make this qualitative result more quantitative, and that is through the use of a colorimeter. Now, a colorimeter is a piece of equipment that scientists use to measure the amount of light that is either absorbed by the solution or transmitted through the solution. Colorimetry works on the principle of the Beer-Lambert law. Now, this law says that the concentration is directly proportional to the amount of light that's absorbed or transmitted. This means that the concentration will increase as absorbance increases or as transmittance increases. Let's take a clear test tube as an example. So when I pass light through this test tube, all the light will move through the test tube and none will be absorbed, meaning that it will be 100% transmittance and a 0% absorbance. If I increase the color of the test tube or solution slightly, then some of the light will be absorbed by the test tube solution, but there would still be some light being transmitted. Remember, the reason why we see color is because the light spectrum of light is being absorbed by an item, and only the color that we see is being reflected, and that is why we see that color. Now, in this example, 40% of the light was absorbed and 60% transmitted. But if we increase the solution's color even more, then even more light will be absorbed by the solution and less light would be transmitted. So in this example, 100% of the light was absorbed and 0% transmitted. If we look at opposite ends of the light spectrum, if we see a white object, it means that all of the light is being reflected back to our eyes. But black, being the darkest color, means that all of the light is being absorbed, and black means the absence of light. So as the color intensity increases or darkens, it means the absorbance increases and the transmittance decreases. So we can add a numerical value to this color change to determine the rate of reaction of the enzyme. So how do we apply this to our amylase enzyme experiment? So the first thing that we're going to do is we're going to calibrate our colorimeter. So we're going to use a weak iodine solution as an endpoint. This means that we'll calibrate it as 100% transmission. This will show that no color change has occurred from this point. We're then going to add a red color filter. This will make our results more accurate and more precise because we will only be using one wavelength of light. This specific wavelength, meaning red, is chosen because it will be totally absorbed when the blue-black color is present. Remember, the reason why we see the blue-black color is the blue-black color is being reflected towards our eye, meaning red will be absorbed. We will then take the results and then graph them. So if we look at a transmission over the percentage starch, as our starch percentage increases, our transmission will decrease because the color from our solution will go from a weak iodine color, which is a yellow color, to a dark blue color, meaning more of the light is being absorbed and less is being transmitted. But if we look at a percentage absorbance over the percentage starch graph, then our graph will have an increase in gradient, meaning that as the percentage starch increases, our absorbance increases as well because the color becomes darker and more light is being absorbed by the solution. There are several different factors that can affect the rate at which an enzyme is active. The first one is temperature. Now, if we look at the tertiary structure of a polypeptide, we can see that it's held together by several different bonds. These can include ionic bonds, hydrogen bonds, and even disulfide bonds. Now, when the temperature is too high in an enzyme reaction, these bonds can break. And when these bonds break, it changes the enzyme's active site's tertiary structure. This will mean that the substrate will not be able to fit into the active site and no product will be able to form. But the change in temperature isn't all bad. If we look at a graph of the activity of an enzyme over the temperature of the solution, we can see that as the temperature increases, the activity increases as well. This is because of the kinetic model theory. So as the temperature increases, our kinetic energy increases, meaning that the particles will move faster, meaning more substrate will collide with enzymes and more substrate will be turned into products. But we do get to a point where we have the optimum temperature, and this is the temperature at which the activity is at its highest. Past this temperature, the activity decreases significantly. This is because the bonds that keep the active site together are broken and the active site loses its tertiary structure and the enzyme will be denatured. The denaturization on an enzyme is irreversible, meaning that we will not be able to get the shape of the enzyme back to its original shape, and the activity will stay decreased until more enzyme is added. The next factor that affects the activity of an enzyme is the pH. So let's look at the graph of the enzyme activity over the pH changes. So we can see as the pH increases, there's an increase in activity and then the same decrease in activity as well. So we have an optimum pH, which is the pH at which the enzyme activity is at its highest. Now, when the pH deviates slightly from the optimum pH, the acid will release hydrogen ions and the bases will release hydroxide ions. Now, these hydrogen and hydroxide ions can break the hydrogen and ionic bonds in enzymes by bonding with these bonds. This means that the shape of the enzyme active site will slightly distort. This is a reversible change and can be put back together. But if the pH differs majorly, then more of these hydrogen and hydroxide ions will break more bonds and the change of the active site will change completely and the enzyme will denature. This is an irreversible reaction. But depending on the enzyme, there are different optimum pH and different enzyme activity across pH graphs for each enzyme. For example, pepsin in the stomach has a very acidic optimum pH, whereas protease in the small intestine has a much higher, more alkaline optimum pH. Now, when doing enzyme activity reaction experiments, we have to remember to keep the pH constant and at optimum pH to achieve the highest enzyme activity. This can be achieved by using a buffer solution. Now, a buffer solution is a solution that would maintain the pH at a certain pH no matter what activity or what reaction is taking place. Next, we have the enzyme concentration. Now, let's imagine we have one enzyme in a solution. That would mean only one substrate molecule can be turned into a product. But if we double that and have two enzymes, two substrate molecules can be turned into two products. Double that again and our products double as well. So in this way, we can see that as we increase the enzyme concentration, we'll increase the rate of the reaction. So we can look at the graph of the rate of reaction across the enzyme concentration and see that the increase is constant. This will only be if there's an excess of substrate available for the enzyme to react with. But if there isn't enough substrate, meaning that the substrate's limiting, our graph will plateau at the end. So when the graph is increasing, we know that our enzyme is the limiting factor. But when our graph plateaus, our substrate is the limiting factor, meaning that there isn't enough substrate to achieve the maximum rate of reaction. We then have the substrate concentration that can affect the activity of an enzyme-controlled reaction. Now, just like in enzyme concentration, the substrate concentration increases the rate of activity as the substrate concentration increases. So let's say we have a certain amount of enzyme in our solution. If we only have one piece of substrate, only one product will fall. But if we have more substrate, more product will fall. But this is also only up to a certain point. If we have excess substrate and only a certain amount of enzyme, only that certain amount of substrate will be formed into a product. So the rate of reaction graph over substrate concentration will increase and then plateau. So as the graph increases, it means that the substrate concentration is the limiting factor. That's the factor keeping the reaction back. But when it plateaus, it's the enzyme concentration that's the limiting factor. So with this, you have to make sure what your graph represents and where the limiting factors lie. After we are able to construct a graph of the rate of reaction over the substrate concentration, we can use the Michaelis-Menten constant to investigate the affinity of the enzyme to the substrate. So when I say infinity, it means what is the likelihood that the substrate and the enzyme will form an enzyme-substrate complex and that the reaction will occur. The reason why this is important is because when we discussed the induced fit model of enzyme reactions, we did say that different substrates are able to fit into the same enzyme. Different molecules that have similar shapes to the substrate can also fit into the same enzyme, but the enzyme will have a higher affinity to certain substrates than to others. We represent the Michaelis-Menten constant by Km. So if it has a high Km, it means that the enzyme has a low affinity for that specific substrate. And if it has a low Km, it means that the enzyme has a high affinity for that specific substrate. To measure the Michaelis-Menten constant, we'll have to find the initial rate of reaction. Now, the reason why we measure the initial rate of reaction is because the initial rate of reaction would be the highest for a reaction at a specific concentration of substrate. Remember, as the substrate is being used up, the rate of reaction will start to drop. So the first part of the reaction would be the fastest. So once we find the initial rate of reaction for different substrate concentrations, we can graph them. And our graph would look something like this. So it would increase as the substrate concentration increases and then plateau at the end of the graph. This is where my substrate concentration is no longer limiting, and we call this the V max. So this is my highest rate of reaction or highest velocity that can be achieved. Let's say this is 10. Now, to get Km, we'll take half Vmax, so we'll get 5. We'll draw a line until it touches the graph and then go down to my substrate concentration. Now, my substrate concentration is my Michaelis-Menten constant. So it's not the initial rate of the reaction, but rather the substrate concentration for half of Vmax. Now, this is measured in millimoles per decimeter cubed. Now, the last factor that can influence my rate of reaction is inhibitors, and we get two distinct types of inhibitors. We get competitive inhibitors and non-competitive inhibitors. Let's say we have an enzyme. So far, we've learned that an enzyme has a specific site called the active site where a substrate can bind to for the reaction to occur. But enzymes also have a different specific site called the allosteric site. This is where some inhibitors can bind. Now, in competitive inhibitors, the inhibitors won't bind to the allosteric site. Rather, they would move to the active site and bind to the active site, taking up space in the active site, meaning that the substrate won't be able to bind to the active site and no reaction will occur. Now, one thing about competitive inhibitors is as we increase the substrate concentration, the activity of the competitive inhibitor is reduced and my rate of reaction will increase again. In non-competitive inhibitors, my inhibitor will bind to the allosteric site. This will distort the active site and the enzyme will no longer be complementary to the active site, meaning that it won't fit into the active site and no reaction will occur. Now, unlike competitive inhibitors, non-competitive inhibitors cannot be counteracted by increasing the substrate concentration. Let's compare the activity of an enzyme without an inhibitor and then with both competitive and non-competitive inhibitors. So we will compare this as the substrate concentration increases. So for a normal reaction, as the substrate concentration increases, the rate of reaction will increase and then kind of plateau at the end where the substrate concentration is no longer limiting, but another factor is limiting. For a competitive inhibitor, the rate of reaction will also increase as the substrate concentration increases, but the rate of reaction will increase at a lower rate. As the substrate concentration increases, the action of the competitive inhibitor will be overcome by the substrate concentration and it will still increase up to the normal enzyme rate. Now, for a non-competitive inhibitor, we can see that although there's still an increase in the rate of reaction as the substrate concentration increases, it's much lower and the reaction plateaus much quicker than a normal or competitive inhibitor reaction. We can see that increasing the substrate concentration does not affect the action of the non-competitive inhibitor.